Okay guys, let's talk a little bit about crossing over. And I use a different method of video here. I got a little bit of 4-point on the side because I wanted to make sure we understand what's happening. Similarly to your karyotype, it's important to realize, think about your karyotype once again. You will have homologous chromosomes next to each other, right? And think about what it means. What are those copies from? Since now you understand meiosis, and you know that at the end of meiosis you get gametes that are haploid, so they will have only one copy of each of homologous chromosomes, right? You know that to make a full individual, you need to merge with another gamete that has also only one copy of each. So you make pairs of homologous chromosomes. That's the whole fertilization. That's the whole thing. We forming homologous pairs. And we need, like, there's also a really good thing about sexual reproduction when you do that process. It's just, instead of just making a clone, is you create a lot of variation. But how is that variation coming about? Well, crossing over is one key to variation creation. So it's not creating new alleles. Remember, that's the only process. The only process that creates new allele is mutation, one of the 4 evolutionary processes: mutation, genetic drift, natural selection, and migration. So remember that as well. The crossing over is when some of the information of one chromosome from the homologous pair goes on the other one, as you can see down here on the picture. So let's talk a little bit about those chromosomes, why we're drawing them in those colors. And I'm sorry about that. It's not really work, but it helps us to understand things. When we would draw a karyotype, we could actually use the same color, which is blue and red. I say it's not really work because one is pink, actually. They use in the PowerPoint. One is from the mom, one is from the dad. So it just means that each of those homologous chromosomes come from different organisms initially. So we're going to call the blue the parental, the father copy, and the pink the mother's copy. So those chromosomes, if you focus as they do in that PowerPoint, they start from one chromosome of a pair, that is from the dad, and one chromosome of a pair, the other homologous chromosome, that's from the mom, right? And that here is unreplicated. So as they show in that PowerPoint, if you want to receive an email from Riley, the student from another class. So if you see on the PowerPoint, you see those letters A, B, C, D, E, F. I'm just going to do those like that. And for the mother, it's all recessive. It's A, B, C, D, E, F. When they replicate, you make a new molecule of DNA. Remember, so it's the same letters because you just copy the DNA, but do remember that although the color is the same, it's a semi identical replication format. So that molecule actually split in half, and one half is creating a new one. So it's actually half and half, new and old. And that one gets also replicated. And all those letters, they represent different alleles, but they, by being A, it's one gene, the gene A, the gene B, the gene C, D, E, F, because they have the same location. Now the alleles are capitalized or non-capitalized letter. Now that's Sheen. It's actually someone from your class. So now that we know that, the crossing over occurs when one leg of one chromosome switch over and cross with the other homologous chromosome. When would that happen? That can only happen during prophase 1. Why? Because in prophase 2, the homologous chromosome have been separated already. You're going to separate the sister chromatids. So in prophase 2, those two are in different cells already. In prophase 1, they're still in the same cell, and they haven't been separated yet by anaphase 1. But in prophase 1, they condense, they appear in the nucleus, the nuclear envelope disappears, the spindle fibers form, the centrosomes are building up those spindle fibers. And what can happen is, as you can see here, the leg cross. That's why it's called crossing over. And during that prophase, when the leg cross, since they coincide, they are homologous chromosomes, they carry the same gene. That means that the letter ABC here is also ABC there. The sequence is different, but what can happen is the entire part of that leg going to be switched. And that will make a mix up a little bit the alleles and the combinations that are possible. Because now we're not looking at something that would be so predictable as we had before. Because without crossing over, you actually can always predict what's going to be in your gametes. Whereas with crossing over, you don't know. That's why traits get mixed up. That's why parental traits coming from mother or father get completely mixed up because of those crossing overs. And when you, as you can see in that PowerPoint here, you can see how that carries on, and the gametes are different than what we would expect. If you would have expected normal gametes without crossing over, you would have blue, blue, pink, pink, and here you have blue, pink, blue, blue, pink, pink. So it's non-expected, and that's what the crossing over is all about. And it's really prophase one. Crossing over is creating diversity. It's one of the big advantage of sexual reproduction. It's really a phenomenon. It's pretty unique. I just wanted to make sure we knew about the coloration: blue from one parent, pink or red from the other parent. And then that's how they get mixed up, and that's how your gamete set is different. Now keep in mind, I'm showing you one chromosome set here from one homologous pair. You have 23 homologous pairs in humans. Crossing overs happens all the time. They happen all the time in prophase one. And now when you make gametes, imagine you make sperm cells, each of those turn into a sperm cell, or one of those turn into an egg, and the other one in satellite cells for that egg. You do that a lot of time. A human female has thousands of eggs. A male produces millions of sperms. And the crossing overs happen all the time. So it's a big, huge lottery. So that's really, really the beauty of that crossing over. So a little follow-up of the crossing over part of the video. I wanted to talk about independent assortment really quick. So keep in mind those colors I just talked about, pink and blue, and you can see them here, how pink and blue. So if you think of a cell during the separation of homologous chromosomes, so anaphase 1, right? Well, actually, anaphase 1 is a good one. It's when the homologous chromosomes separate, but the PowerPoint here shows you the process of starting with metaphase 1 here. So if we do metaphase 1, when the homologous chromosomes align, and we use the same color code we had before when you have the blue and the pink. Think about it in 3D. If we think about this like there's just one pair of chromosomes, remember the A, B, C, D, E, F, and the A, B, C, D, E, F that we just did. Well, if we look at those chromosomes, they can, when you have only one pair, they can align like that, right? And they will be pulled apart by the centrosomes on either side. It's just something to remember about metaphase 1 is the spindle fibers are attached to each of the chromosomes from the pair. But now when you have more than one, as you can see here in the picture, what happens is they can align this way. They can have all one parent chromosomes from the homologous pairs on the top and all of the other parents on the bottom. But it doesn't have to be that way, right? You can arrange different ways. And with 3 pairs, you have already 8 possible gametes. So now imagine you have 23 pairs. Do 23, do the calculation, it's a ton of different gametes. And here we haven't layered on the crossing over. So really the key is to understand that a cell is a 3D object. And the alignment doesn't have to be all one parent chromosomes on the top from the homologous pairs and the other on the bottom. They can be switched. So it could be pair 1, it's this way, pair 2, it's the other way, pair 3, 4, 5, it's the same way as pair 1. You see the idea. It's really the way they orient in space. The spindle fibers then grab onto them and then pull apart in anaphase 1. So that's the independent assortment. It means that each pair independently assort during metaphase 1. Each pair of chromosomes independently send one of the homologous chromosome on either side. It's not because pair 1 did the blue top, the pink below, that it means anything for the others. Each pair independently assorts during metaphase 1. Okay, for the last minute, I just wanted to quickly talk about the error that I mentioned in my meiosis video. During meiosis, you can have a non-disjunction, and let me try to make that a little bit bigger here. The non-disjunction occurs when the homologous chromosome, for example, don't separate. So this is only one pair represented, but think about the 23 pairs. You can have only one pair that doesn't disjunct or segregate correctly. So one of the two daughter cells at the end of meiosis 1 has two of the homologous pair compared to the other one doesn't have any. That one has the rest of the chromosomes, like one of each other pairs, and there are 22 over chromosomes in there. So now that cell that's 24 is this one 21 instead of 23 each. And then down the line, meiosis 2, you make gametes that are haploid, but you see how they have two copies. So they semi-haploid, it's weird, and then don't. So when you make the zygote after fertilization, you see how you don't recover two homologous chromosomes for each pair. You have that weird trisomy. And the other non-disjunction can also take place during anaphase 2, when anaphase 1 everything went well, a homologous chromosome are separated, but in one of the two daughter cells from meiosis 1, the non-disjunction of the sister chromatids now take place, so the sister chromatid of the homologous chromosome do not separate normally. And so that's when you end up with, once again, too many chromosomes in one daughter cell, not enough here. So it would be like 24 here, 21 here for human. And those continue with 23. And here we go. That's an that is a non-disjunction.